Cells rule. Sensory epithelial cells. Photoreceptors. Horizontal cells. Bipolar cells. Amacrine cells. Ganglion cells.
Hey Angela, what are you up to?
I'm just looking at that eye model from class, because we have that quiz on Friday, right?
Yeah, the quiz on the retina.
Yeah. So where is the retina on here?
Let's take a look. The retina is the back lining of the eye. So if we open it up from the inside, you can see all of this is the retina. The retina is about 0.5 millimeters thick and contains sensory epithelial cells. The orange and blue are just resembling the blood vessels. Do you see it now?
Oh yeah, I see it. Nice. Hey guys, hi, this is Makai. Are you guys studying for the quiz later this week?
Yep.
Remember, you need to know the function of the photoreceptors, the ganglion cells, the bipolar cells, the horizontal cells, the amacrine cells, and how everything fits together. But I'm sure you guys will ace it. Oh yeah, we know our stuff. Good luck. Thanks. And the fifth type of sensory epithelial cell is the amacrine cell. And you can see here in this diagram, all of the cells work together to transport information to the brain.
Hey everyone, thanks for tuning in to This Week on the Science Classroom. We're learning about the retina this week, and I'm going to start by talking about the photoreceptor cells. These specialized cells perform transduction, which is the process of converting light into chemical signals. They are found in the outer nuclear layer of the retina. As you can see in this image, these are all of the layers of the retina. The photoreceptors are these three cells. They're found in the outer nuclear layer, which is along here. There are two types of photoreceptors in the human eye, known as rods and cones. These are the rods, that is a cone. They have tightly packed membranes containing the photopigment rhodopsin, which is needed for the light photons to actually reach the cells. Over here is just a closer picture of the structure of the rods and the cones. This one's the rod, this is the cone. There's the synapse at the bottom, the nucleus, the green are the mitochondria. And the outer segments are what contain the discs, which have the rhodopsin. The rods allow us to see in dark light and are not involved in color vision. They detect the changes from bright to dim light and also detect shape and movement. The rods are the most abundant type, and there are 120 million of these just in the retina. The other type, which are the cones here, there's 6 million in the retina. These are which are active in bright light and allow us to see color and detect the fine details. This is why you can't see colors very well in the dark, as the cones are not activated. There are 3 types of cones, one sensitive to blue wavelength, one sensitive to red wavelength, and one to green wavelength. They are most sensitive to these 3 colors, green, blue, and red. However, we can also observe colors with wavelengths in between these, as we see so many different colors. For a person to actually observe a color, at least 2 of those 3 cones I just mentioned must be triggered. But the color you see depends on how excited the certain cones respond to that wavelength of light. That's all the time we have for now, but come back in and view the next video to do with the retina. Thanks, guys!
Welcome to the Cell Biology Game Show. I'm your show host, Epithelia. Today we have 2 cell biologists in training. Sarah and Kayla, come on out. All right, let's get started. Someone who is colorblind does not have a particular type of cone in the retina, or one type of cone that may be weak. What percentage of males are colorblind? 0.5%! I'm so sorry, Kayla, that is incorrect.
That percentage corresponds to females. Sarah, do you know the answer?
Is it 8%?
Yes! Color blindness is much more common in males than females. All right, next question. What is the region that is very thin and has tightly packed cones, which allows for clear vision? This part only contains cones, no rods.
The fovea?
That is correct. All right, this question requires much more explanation, so it's worth double the points. Are you ready? Why do we have a blind spot?
We have a blind spot because one part of the retina does not contain any photoreceptors. Any image that falls on this part won't be seen. It is the region where the optic nerves come together and exit the eye on the way to the brain.
That is precisely correct. All right. When you first enter a movie theater or a dark room, the cones in the retina are working and the rods aren't yet activated. How long does it take for the rods to get activated?
7 to 10 minutes.
Precisely. All right, so that ends our round 1. Stay tuned for round 2. Hey everyone, welcome back. Thanks for tuning into the Science Classroom. Now, we'll be talking about horizontal, bipolar, and amacrine cells. So let's start off. Bipolar cells provide a direct link between photoreceptor terminals and ganglion cell dendrites. So let me show you. These are the bipolar cells, and like we talked about before, the photoreceptor cells. So right in here. There are 11 different types of bipolar cells in the human retina. 10 are for cones, and 1 are for rods. These bipolar cells are found in the internuclear layer. So right here. Like most cells in the retina, bipolar cells communicate via graded potentials rather than by action potentials. What does that even mean? In cells of many different types, there's something called an electrical potential difference. An electrical potential difference occurs when there is a net separation of charge between two locations, measured in volts, shown here. When we talk about the differences in membrane potential, we call it graded potential. Depending on the stimulus, graded potentials are either hyperpolarizing or depolarizing. Action potentials always are depolarizing and lead to reversal of the membrane potential. Next, horizontal cells are retinal neurons that mediate lateral interactions between photoreceptor terminals and the dendrites of bipolar cells. So right here we have horizontal and bipolar. They regulate signals from photoreceptors in different conditions of light, making transduction more sensitive in bright light and less sensitive in dim light. In terms of structure, horizontal cells have many different subtypes and are very large in relation to most other cells in the nervous system. Their structure can differ from species to species, but one thing that they have in common is they do not look like any ordinary axon. These cells are found in the outer plexiform layer, so right here. Amacrine cells mediate interactions between bipolar cell terminals and the dendrites of ganglion cells. So right here, like I said, bipolar, ganglion, you can see that connection. Varying in many different sizes, there are 20 different types of amacrine cells. They are found in the inner plexiform layer, like I said, and they are known to have many different functions, but a lot of those functions are unknown. Well, that's all we have time for today. See you next time on the Science Classroom.
Time for a fun fact. Dogs have 2 types of cones compared to humans who have 3 in the retina, so dogs' color vision is more limited. They see similar to how humans with red-green color blindness see.
Hey everyone, welcome back. Thanks for tuning back into the Science Classroom. So far, we've covered the photoreceptors, the horizontal cells, the bipolar cells, and the amacrine cells. The last cell in the retina that we're going to be talking about is the ganglion cells. The ganglion cell bodies are found in the ganglion cell layer. Ganglion cells are the only type of cell in the retina that send out neural signals by means of action potentials rather than graded potentials. There are 3 categories of ganglion cells. The first type is type P, which stands for parvocellular. These cells are small and are the most abundant type, making up 90% of the total ganglion cells. The next type is type M, which stands for magnocellular. These cells are larger than type P cells, and they make up 5% of the total ganglion cells. These cells make up the optic nerve and carry information for retinal stimulation to the rest of the central nervous system. The last types of ganglion cells are the non-P and non-M cells, which also account for 5% of the total ganglion cells. These cells are still being researched and are not yet described. So how do all these cells work together? Well, the pathway for the transfer of information to the brain starts with the photoreceptors, who are the first cell to interpret the information. From there, the signal is sent to the bipolar cells with help of the horizontal cells. Once the bipolar cells receive the signal, the information is sent to the ganglion cells with the help of the amacrine cells. From there, the information is sent directly to the brain by the optic nerve where it can be processed. The image that forms on the retina is actually upside down, but the brain processes the data and perceives it right side up, and that's how an image is formed. That's all the time we have today, folks. Thanks for tuning in to the Science Classroom.